Hi, good morning. Thank you, Jose. And everyone, welcome. And thank you for joining us for the GenomeStudio Genotyping Introduction webinar. So I'll go ahead and get started. Our objectives today are to introduce the Illumina GenomeStudio software to new Infinium Genotyping array users, as well as discuss the genotyping module capabilities. We'd like to show you how to perform basic analysis workflow in GenomeStudio 2.0 from project creation to report generation. And before I get started with the GenomeStudio webinar, I'd just like to show you this reference slide showing all the various Infinium webinars that we have available on our training website. So as you can see, we have a couple of methylation-focused webinars and a couple of cytogenetics and CNV analysis webinars. And we also have several webinars that cover genotyping. So if you'd like to learn more about these other topics, go ahead and go to that webpage and watch those other webinars. So today's focus is on the GenomeStudio 2.0 genotyping. So the purpose of genotyping array analysis is to go from a process bead chip to a final report. So you have a scanner that scans the processed arrays and it extracts the raw data in the form of these intensity data or IDAT files. All the subsequent steps afterwards are done in GenomeStudio, and the software is able to normalize data and call genotypes for the samples. And you can also use the software to QC your data and modify the clusters. And finally, the ultimate goal is to generate your output data, which is a final report that contains your allele calls. So what is the GenomeStudio software? So this is just a high-level overview of the multiple capabilities that GenomeStudio is able to perform. So GenomeStudio can create custom cluster files or .egt files to optimize your call rates. And GenomeStudio can be used to perform QC of your samples and also perform copy number variation or CNV analysis. And as I've mentioned, it can create genotype reports in text format. And there are also plugins available that allow you to export reports that can be used for third-party software analysis. So depending on the bead chip that you're using, GenomeStudio contains multiple modules that can support multiple analysis methods. So today I'll be focusing on the genotyping module, and this module allows you to generate genotypes for your samples and perform CNV analysis. But in addition, GenomeStudio also has a polyploid genotyping module that allows you to analyze data from polyploid organisms. And GenomeStudio also has a methylation module to analyze methylation data. And although Illumina no longer produces gene expression arrays, if you do have data from those arrays, they're able to be analyzed in GenomeStudio as well. So which GenomeStudio software should you use? So if you are using a genotyping or cytogenetic array, we recommend using GenomeStudio 2.0. So even though both GenomeStudio 2011, which is the older version, and GenomeStudio 2.0, which is the newer version, both have a genotyping module, we recommend using GenomeStudio 2.0 because it uses the latest GenTrain 3.0 clustering algorithm, which is a more improved algorithm compared to the older one, which is used by GenomeStudio 2011. So in short, if you have a genotyping or cytogenetic project and you want to perform genotyping analysis, we recommend 2.0. But if you have a methylation array, definitely go for GenomeStudio 2011, which contains the methylation module and the gene expression module as well. In addition to GenomeStudio, we also have two other software that are available for analysis of Infinium data. So Beeline is a software that can be used for high throughput genotyping data analysis, and Bluefuse multi-software can be used for copy number variation analysis. And if you refer back to the list of webinars on the previous slide, you can see that there is a separate webinar for each of these software options. So again, I mentioned that there are 2 different versions of GenomeStudio. So you can go forward, but you can't go back. So if you have projects that are created in GenomeStudio 2.0, they cannot be opened in the older GenomeStudio 2011 version. But if you do have projects that were created with a GenomeStudio 2011, they can be opened in GenomeStudio 2.0. So in general, if you do have a genotyping project, we recommend GenomeStudio 2.0 so you can take advantage of the improved features and clustering algorithm and performance of that software. So now that you're ready and you know that you want to have or to analyze your data using GenomeStudio 2.0, here are the minimum system requirements that you want to make sure that your computer meets. So you want to make sure your computer is running a Windows 7 or higher operating system, and that it has a 64-bit processor, a minimum of 8 gigabytes of RAM, and 250 gigabytes of hard drive storage space. So this software is available at no charge from the Illumina website, and there's a link below, or you can just search the Illumina website for GenomeStudio software. Once you download it, you can install the software. You simply extract the zip file, and then you run the GenomeStudio installer.exe. And one thing I would like to note is that make sure you just select the default file paths so that the software knows where to write the files. So for the rest of the webinar, I'll be describing the GenomeStudio workflow in more detail, and there are several steps. So first, you want to create a project, and then steps 2, 3, and 4 are considered optional, and they're data QC steps. And even though they're optional, we do highly recommend performing these steps so that you can make sure you're getting accurate genotype calls. So these intermediate steps involve evaluating the controls, then evaluating and filtering your samples, and then evaluating and filtering the SNPs. And then finally, at the end, you're ready to export your reports, and you'll get your allele calls in a text format. So first, I'll go over how to create a project in GenomeStudio. So you have your scanner, which scans the process bead chip, and it generates red and green IDAT files for each channel. And the IDAT files contain the XY location and intensity data. And this is a required file. And in addition, another required file is a bead pool manifest or .bpm file. And this contains the bead ID and sequence information. So basically what GenomeStudio does is it combines the intensity values from the IDATs and correlates it with the probe information in the bead pool manifest. So in addition to these required files, there are also optional files, but files that you might find useful in addition to your required files. So one optional file is a sample sheet. This is a CSV file that contains sample information. For example, if you have replicate information or gender information or sample groups, you can include that information in the sample sheet, and this will be imported into the project. Another optional file that is very helpful is the cluster file or the .egt file, and these contain the genotyping cluster positions. So loading this file will help speed up the project creation process because the software will already have the cluster positions in the EGT file rather than having to calculate it. So once you have these files ready and they're imported, GenomeStudio creates a workspace, and this is a BSC file. So this slide is mostly just for your reference, again, describing what each of the required and optional files are. One thing I'd like to point out is that depending on the bead chip type that you have, this will determine where you can access the files. So for a commercially available file, you can download a manifest and cluster file on the product-specific support page. However, if you have a consortium or custom bead chip, you can download the My Illumina the files from the MyIllumina custom product file search. However, there is no cluster file available, so you will have to generate a custom cluster file in GenomeStudio. And finally, for the sample sheet, there are templates available under the support files for the specific B chip type, so you can download those templates and work from that. So now that I've discussed the required and optional files, we're ready to create a project in GenomeStudio. So when you first initialize the software, you'll see this screen very briefly, and then it will take you to this GenomeStudio launch page. And you'll have two options for creating a project. You can either select File, New Project, Genotyping, or you can double-click this Genotyping button under the New Project. Either way, it will take you to this project creation wizard. And when you see this project creation wizard, it will just take you step by step through the process. So you go ahead and click Next, and then it'll take you to a screen where it asks you to select a folder where you want to save your files, and this will be the repository. And it'll also prompt you to enter a name for your project. So go ahead and click Create and then type in your project name. Next, you'll be given two options of whether you want to use a sample sheet to load your data or whether you want to load intensities by selecting folders with intensity files. So I'll go over the first option, which is using the sample sheet to load sample intensities. But before I do that, I just want to make a side note that one suggestion I would have for best practices is to combine all the required and optional files into one repository folder so that you know where all your files are stored, so that when the software prompts you to navigate to the files, you'll know exactly where they are. So this is just considered a best practice, but it's up to you how you want to store your files if you know where elsewhere they're located in the computer. So for this particular example, I have the manifest, cluster file, and sample sheet all in one folder. And then I have an IDATS folder, which is the parent folder that contains all the individual scan folders. Another thing that I'd like to go over is the sample sheet guidelines. So you want to make sure that your sample sheet is formatted correctly so that GenomeStudio can import the information properly. Otherwise, if it's not formatted correctly, GenomeStudio will throw an error. So I'll go over these in more detail in the next few slides. So as I've mentioned, the sample sheet templates are available under the product-specific support file section. So in this example, I'm using a global screening array version 3.0 sample sheet, which I downloaded from the website. And this is what the file looks like. It is a CSV file, and you can see it contains the sample information. And another thing is that under column B, it contains the exact name of the manifest, which is a GSA 24V30A1. So you want to make sure that the manifest in this field exactly matches the name in the .bpm file, because the software uses this information to access the correct file. Another thing to note about the sample sheet is that you want to make sure the barcodes are formatted correctly. So you can select the barcodes and then format them to remove the decimal places. And this ensures that the full 12-digit barcode is displayed and it's not concatenated. Also, you want to make sure that all the required columns are filled in. So for a genotyping sample sheet, the first three columns are required. So this is the sample ID, the Centrix barcode, and the Centrix position. So you want to have the manifest name there, and you want to keep that letter A there as well. So the A references the Centrix barcode A and the Centrix position A. So once you have your sample sheet ready, another thing you want to make sure is that every time you make edits to your sample sheet, that you have to format the numbers and you have to save it as a CSV file. You also want to make sure to close the file before you load it into GenomeStudio. So now that you have your files ready, you can navigate and browse to your sample sheet, as well as your data repository, which is where your IDATs are saved, and the manifest repository, which is the folder that contains your manifest. So now we'll go over the other option. So instead of using a sample sheet, if you choose not to use a sample sheet and you just want to point to a particular location in your computer, you can select this second option, which is Load Sample Intensities, by selecting directories with intensity files. So as a reminder, you can see here in the small picture that this is the repository organization. So I have all the files in one location so that it's easy to navigate to that. So it'll take you to the next screen where you can navigate to the SNP manifest BPM file, as well as the data repository, which is that parent folder that contains the barcode folders. So once you select that folder, it will pull up all the available scan folders in there, and you can select to add one or multiple folders, and they will be added to the selected directories on the right side of the project creation wizard screen. And finally, the last step of the project creation wizard is to import cluster positions. So cluster file, again, is not required for project creation, but if you have one available, you can check that box that says import cluster positions and navigate to your EGT file. So all the other options are off by default. So when you're finished, you can click finish, and here I'll show you what it looks like to create a project with a sample sheet. So you create a new project using the option, and then you select a folder where you want your project to be saved, and you type in the project name, and then you click next. And so we're using the sample sheet option this time. So for sample sheet, you browse to the actual CSV file. The data repository is the parent folder, which is in this example, the IDATs folder that contains all the barcode folders. And the manifest repository is the folder that contains your manifest file. So once you're ready, click next. And because we have a cluster file available, we'll select that EGT file from the computer. Click open, and then leave all the other options off by default and click finish. Then you'll see that the project will begin to load, and it'll show you this progress status, and the log window on the bottom will show you the status as well. This is an example of how to create a project without a sample sheet. So the beginning part is the same. You create a project and go to the project wizard. Again, you select your repository and type in your project name. And then instead of the first one, we pick the second option to just load the sample intensities. We select the .bpm manifest file and the data repository, which is the IDATs folder, the parent folder containing the scan folders. And when you select that, you can see that all these folders pop up, and you can select one or all of them, and they'll show up in the selected directories when you click Add. Then when you're in the final window, you select the cluster file and click Finish. And again, the progress status window will appear, and the log window will show you the progress of the project creation. So once your samples are loaded, this window will pop up in GenomeStudio, and it will ask you, do you wish to update SNP statistics for all SNPs? And we recommend that you select no at this point. So you can update SNP statistics later, but in the interest of time and getting your samples loaded in the project, it's better to select no so that you can make your QC evaluations before you update SNP statistics again. And once your samples are loaded, here is just a quick overview of what a GenomeStudio 2.0 workspace looks like. There are several windows with different functions. The upper left is a SNP graph window, and it shows you the cluster positions of the selected SNP. The samples table on the left shows you all the samples that are in the project, as well as the call rate and additional sample information in the other columns. The data table in the center is actually 3 different tables, and you can toggle depending on which tab you want to select. The 3 tabs available here are the full data table, the SNP table, and the paired samples table. So in the data table, every row represents a SNP locus, and the full data table actually shows the samples as well. In the right is the project window, and this shows you which manifest was used to create the project, and this indicates what kind of B chip was used. And it also shows you the data that was imported into the project. The errors table right underneath the data table shows you errors if you have information about replicates and parent-parent-child relationships in your data set. And in the bottom is the log window. As I mentioned before, it can show you the status or the progress of your project. So if you apply any analyses, it'll show you whether that is still in progress or if it's completed. So now that you have your project loaded, you're ready to go through the QC steps. And so the first QC step here is to evaluate the controls. And to do that, you want to review the GenomeStudio controls dashboard. So you'll go to Analysis and select View Controls Dashboard, and this will pull up a window with a series of different graphs. So there's going to be a graph for each channel. There's going to be a red channel and a green channel. And here I'll help you interpret what the graph means. So there is one graph for each channel for each control, and each column represents a sample. And the samples are presented in the same order as in the samples table. So if you ever change the order in the table, it will rearrange things in the controls dashboard as well. And every dot represents a control probe, and each control probe is shown in the legend. So in this example, the purple and the red represent the A's and the T's, and the green and the blue represent C and G control. So these controls are designed to check for processing and sample input, so it allows you to evaluate assay and sample performance. So the sample-independent controls are informative regardless of species, and the sample-dependent controls are useful if you are running human samples. We have a couple of training materials here. There is a Evaluation of Infinium Genotyping Assay Controls webinar and online training course if you'd like more details on how to evaluate these controls. So again, the controls dashboard is useful because it provides information on assay performance and sample quality. And we recommend you check out the Evaluating Infinium Assay Controls webinar, as well as the Best Practices webinar. Before I go into explaining how to evaluate your sample and SNPs, what I'd like to do is provide an overview of the sample and SNP metrics, and I'd like to define these so that you can better understand why it's important to evaluate these metrics. So there are several metrics available for review in GenomeStudio. In terms of sample metrics, the main metric that we look at is the call rate, which is the percent of loci that a genotype was called for the sample. Another sample metric is the log-R deviation, or log-R dev, and this is the deviation in the normalized intensity value. And it can be used to indicate contamination in the sample, And this metric is also used when performing CNV analysis. So for a standard Infinium product, the expected call rate is over 99% average. So in addition to sample metrics, you also want to look at SNP metrics. And one SNP metric is call frequency, and it is the percentage of samples that a genotype was called for the SNP. Another SNP metric is a GenTrain score, and this is a metric that reflects how well GenomeStudio was able to identify groups of alleles in a training data set. So I'll explain GenTrain score in more detail in later slides. And in addition to these two metrics, there are also other metrics available in the data table that a user can review and determine the cutoffs and thresholds. So how exactly are genotypes called in GenomeStudio to be able to arrive at these metrics? So as I've described before, an Illumina array scanner detects the red and green fluorescence intensities for each SNP locus, and this produces genotyping data. So the scanner produces these intensity data files, or IDAT files. Once the IDATs are imported into GenomeStudio, the software can then convert the A and B color channel signals into genotype calls. So to describe this further, signals that are generated in the red and the green channels are proportional to the allele A and allele B genotypes present in a sample for a given biallelic SNP. So looking at this graph, the genotype AA, the signal comes from all A channel. Whereas for genotype BB, the signal comes from all B channel. And genotype AB, the signal comes from both A and the B channels. So theta is another value that GenomeStudio looks at, and this is the angle on which the data points fall. So this is defined so that when theta is 0 at 0 degrees, this corresponds to the AA cluster of samples. When theta is 0.5 at 45 degrees, this corresponds to the AB cluster. And then when theta is 1 at 90 degrees, this corresponds to the BB group. So to make the data easier to interpret and to simplify clustering, GenomeStudio actually converts the normalized intensity, A and B graphs, from the Cartesian coordinates to the polar coordinates. So if you look at the graph on the left, these represent signal intensities for 96 samples at one SNP locus. So in the left graph, the x-axis is the A signal intensity and the y-axis is the B signal intensity. GenomeStudio then takes the theta information and the distance from the x and the y-axis, and it converts it into polar coordinates. And what you get is a graph where the x-axis represents the theta, or the angle from the x-axis, and the y-axis then represents a value called R, which is the sum of the A and B signals. So the purpose of converting or transforming from the Cartesian to polar coordinates is to make it easier to visualize the data points horizontally. So it's much easier to tell that your AA clusters on the left, your AB clusters in the middle, and your BB clusters on the far right. GenomeStudio also uses algorithms that are built into the software to create clusters and assign genotypes. The clustering algorithm is called GenTrain, and this determines the location and shape of each genotype cluster. Another algorithm is the GenCall algorithm, and it determines which genotype bin each sample data point belongs to. So in this example, the GenTrain algorithm was able to determine where the AA, AB, and BB clusters are, whereas the GenCall algorithm will determine where the data points fall. So now that I've explained what these metrics are and how the genotypes are called, you're ready to evaluate and filter your samples. So the main sample metric is the call rate, which is defined as the percentage of all SNPs and indels that are successfully genotyped for a given sample. And this is expressed as an equation where it's the number of called markers divided by number of all markers. And the call rate is a good starting point to QC your assay and sample performance because it's a simple number that will tell you whether it meets a threshold or not. So if you're using a cluster file, like in our example, you can go ahead and proceed immediately to calculating call rates after you make your project. But if you don't have a cluster file available, for example, if you're using a custom or consortium B chip, you want to use the tools in GenomeStudio to cluster your project. And so there are more directions in the Creating Custom Cluster Files webinar if you'd like to create a custom cluster file for your project. So to calculate call rates, first you go to your project, and initially when it loads, you'll see that the call rate is all 0 and it's highlighted in red. And what you need to do is select the calculator button on the upper left right under the samples table name. And once you click that calculate button, the software will calculate the call rates and it will provide you with the call rate values next to the sample IDs. There's also other ways to visualize call rates. You can select this line plot icon, which is in the samples table in one of the series of icons. And when you select that, you can plot call rate on the right axis and P50 green and P50 red intensities on the left axis. And you can see here that there is some variability in the intensities of the samples, but the call rates are generally good. So you can use this information to see whether low call rates may be correlated with low sample intensities. And another way to evaluate your call rates is you can sort the samples table by call rate. And so again, from the previous slide, I've shown you that when you click that calculator button, it'll present you with the call rates for each of your samples. And this information is useful because if you use the call rate specifications, you can take that number and you can exclude any samples that don't meet that cutoff. So for example, for a commercial array, if a sample has less than 99% call rate, you can choose to exclude that. If you are using a custom array, you can set your own cutoff for what you would choose to exclude. And once these samples are excluded, they will be excluded from SNP statistics calculations, and you also have the option to exclude them in your final report. So after you evaluated and filtered your samples, you're ready to evaluate and filter your SNPs. So far we've only focused on the samples table and looking at whether your samples meet the metrics. However, there is also the data table, which contains SNP information. And it's important to review the SNPs so that you can make sure your SNPs are performing well. So in the data table, there is the full data table tab and the SNP table tab. And each row, again, represents a particular SNP locus. So the software runs the GenTrain algorithm to determine the clusters. And when it does this, it assigns a GenTrain score to each SNP. So here are some examples. So on the left is a high GenTrain score for a particular SNP. And an ideal GenTrain score would have 3 clusters where you have equal R at 0, 0.5, and 1 norm theta. So these show ideal cluster positions and distribution. However, if you have a SNP with a low GenTrain score, it does not meet the high GenTrain score criteria. And you can see on the example on the right that even though there are 3 clusters, they're not in the ideal position. So anytime the clusters get broader or narrower or they're not in these ideal positions, the SNP is assigned a low GenTrain score. So in addition to the GenTrain, there's also the GenCall Score, which is a metric calculated for each data point, and it measures how well a sample fits into a cluster. The GenCall score is dependent on the GenTrain score, and it also ranges from 0 to 1. So for this example, a sample with a high GenTrain score will lie near the center of the cluster, whereas a sample that's far away from the cluster has a low GenCall score and no genotype is given. And this shaded area actually represents the GenCall threshold for calling genotypes. So if a sample is not necessarily within the cluster, but it's within this darker shaded area, the software can still call a genotype for that sample. Whereas you can see in the black dot that if it falls outside that shaded area, no genotype is given. So to reiterate the difference, because these do sound very similar, a GenTrain score is SNP specific. So it is presented as GenTrain score in the full data table and the SNP table. The GenCall score is SNP and sample specific, and it's presented as score right underneath the specific sample in the full data table, and score also appears in the samples table. So in addition to the GenTrain and GenCall score, there are also additional SNP metrics that are available for review in the SNP table. And in order to start reviewing this, you should first calculate the SNP statistics. So to do this, you would go to Analysis and then select Update SNP Statistics. Then you'll be ready to review your SNP metrics. This is a reference table showing you some single variable metrics that you might consider reviewing. If you'd like to have more information on these specific metrics and the cutoffs and information, you can consult the Infinium Genotyping Data Analysis Technical Note, which provides a very good summary of how to perform genotyping data analysis in GenomeStudio. So as you're reviewing your SNPs, you might notice that certain SNPs can be improved and modified to improve call frequency. And to do this, you can simply right-click and select cluster selected SNPs, and you can do this in both the full data table or the SNP table. Another thing you can do is in the SNP graph, you can manually move and resize the clusters. So if you're on the SNP graph, you can click Shift on the centroid and you can grab the cluster circle. So in this example, the clusters were generated from a standard cluster file, and you can tell that the data points are not falling in the clusters. So what you can do is you can recluster the SNP locus, and you can see that the clusters have been moved and resized. And after this adjustment, you can get improved call frequency. So that's an example of a SNP that can be improved by reclustering. However, there are certain SNPs that really can't be improved. And in this case, you just want to zero them out. So in the data table, you can right-click and select Zero Selected SNPs, and these SNPs will be zeroed out from your data. So if you'd like more information, you can consult the custom cluster file webinar. But in summary, when you're evaluating SNPs, you can modify some SNPs to optimize clustering, and others you will have to zero if they are poorly performing. And once you're done reviewing your SNPs and you're happy with them, you can actually export a cluster file EGT file, which you can then use for future projects which use the same B-chip type and similar samples. So now that you're finished with evaluating your samples and SNPs, and you've finished QCing the data, you're ready to export your final reports and these will contain your allele calls, which is your ultimate goal. So in order to start exporting your data, you want to go to Analysis, Reports, and then select Report Wizard. It will take you to a report wizard similar to the project creation wizard, which will take you step by step. And in this screen, it gives you the option of selecting a report type. The most popular one is the final report, which contains the allele calls. But if you only want specific information such as statistics for your samples or your loci, you can select the other report types. And if you are interested in exporting reports for third-party software analysis, you can also use the report plugins. So once you reach this screen for the genotyping report wizard, you'll be given the option of selecting what you'd like to include in your final report. So you can select options from the available fields and select show, and they will appear in your report. And you can see there's several options here for how to present your allele information. And you can even change the order. So if you'd like the information to appear sooner in your report, like more towards the left, you can reorganize and rearrange the order of the fields. And you can see there's many available fields on the right. But if you decide you change your mind and you don't want to include certain information, you can just click hide and they'll go back to the available fields. And so you can group your report by sample or SNP, and you can create either a tab or comma delimited file and select the number of samples per file. Then you'll have to choose a folder to save it and enter a report name, and then you can click finish. And it'll give you, it'll export a final report. And so you should be able to view the final report using either Notepad or Excel. So as I mentioned, there are several ways to, or there's several options to export the allele information. So you can select the strand designation in your final report. The default output is the top alleles, and this is based on the Illumina top bot strand designation. Another option is the forward alleles, and this is based on the source strand. So for example, a SNP database source such as dbSNP. Another option is design alleles, which is based on the Illumina strand from which the probe sequence is derived from and designed on. But the most popular option is the plus alleles, which reports from the RevSeq plus strand. And then finally, there is the AB allele option, which reports the alleles as the Illumina AB genotypes, which are the generic designations. So if you'd like more information on the strand designations, there is a strandology webinar that's available on the Illumina website. And there's also report plugins that are available. So for example, if you'd like to export your data and analyze them using Plink, you can use the Plink Input Report plugin, and it will generate files that can be used for input into the Plink software. There's also the CNV Region Report plugin, which allows you to output 3 types of reports. So there's the standard report, the allele-specific copy number report, and the Plink CNV input report. And if you use a CNV region report, this will first require you to run CNV partition plugin before you can create the report. So there's more information on this in the CNV analysis using GenomeStudio or Bluefuse multi-software webinar. And one final note, so after you've exported your report and you're happy with your data, it's important to make sure that you're saving your GenomeStudio project because the project does not autosave. So to save, you can click Control-S or you can go to File and Save Project. If you'd like to share your project with a collaborator, what we recommend is to copy and send the whole folder containing the BSC file as well as the data and the CNV folder. don't just send the BSC file because that does not contain any data. All the data associated with the project is actually contained in that data folder. So also don't change the folder or file names, just keep them the same. And so in this example, all of these files are contained in the GSA v3 test. So if you were to share this with a collaborator, you would share the GSA v3 test folder, and you can zip that and send that to your collaborator. So here are some additional resources now that we've gone through how to create your project and review your data. So if you'd like to download the GenomeStudio software, there is a GenomeStudio support page, and you can download the installer. And the support page also contains documentation if you'd like to download and review the user guide. And these are links to the recorded webinars that I've mentioned throughout the webinar. And there are also a couple of technical notes in the array bulletins that go over Infinium data analysis, as well as how to interpret strand designation and manifest column headings. So if you don't have a GenomeStudio project, but if you'd like to kind of play around with the software, there are demo projects available on the Illumina website. So for example, for the Global Screening Array version 3.0, you can go to the support file section and download the demo data and the sample sheet, and you can get started and load the project and play around with the software and check out all the different functions. So that's definitely one option if you don't have your data yet. And so with that, thank you for taking the time to attend this webinar.